we saw the general characteristics of the prokaryotes, right? This is what we saw. We saw how the prokaryotic organisms, they are very, you can say, primitive organisms, and they do not have a membrane-bound nucleus, nor do they have membrane-bound cell organelles in them. Now, in them, they have a cell wall present, but their cell wall is made up of peptidoglycan. Okay, and these are the organisms that have additional chromosome, like the normal genomic chromosome, the gene they have. Other than that, they have plasmids, which are extra self-replicating chromosomes that they have, or segments of DNA that they have. Depending upon the shape, the bacteria, and you must know that the prokaryotes, the majority members of the prokaryotes are bacteria. So when we talk about the bacteria, so they are of 4 types depending upon the shapes. So if they are coccus, coccus means spherical in shape. Bacillus means rod shape. Spirillum means spirally shaped, and Vibrio means comma shaped. Now, this cell envelope things, have I done this?
No, ma'am.
Okay. Now see, normally when we think about it, see the outermost layer of the cell, like the limiting membrane of the cell, is called as the plasma membrane, which is there in every organism, eukaryotes, prokaryotes, both. Now these eukaryotic organisms, they have 3 layers, and together these 3 layers make their cell envelope. Okay, so outer, middle and the inner layer, like 3 layers together, will be making up their cell envelope. Now their outermost layer is called as the glycocalyx. So the outermost won't be the cell wall or won't be the plasma membrane. It is instead it's going to be the glycocalyx followed by the cell wall, and then the plasma membrane would come. In us, normally plasma membrane is there, nothing else is surrounded. So the outermost layer is the glycocalyx, the middle layer is the cell wall, and the innermost layer is the plasma membrane. Now this layer is very important for the bacteria, because you know that bacteria, they are mostly pathogenic, right? They cause diseases, and because they are present in such extreme conditions also sometimes, so this layer helps them to survive anywhere and everywhere. Okay, so it is a chemically complex layer that is present in most of the prokaryotes, particularly bacterial cells. And it's not that all of them will be acting like separate units. They are present together and they act like a single protective unit, which we call as the whole entire cell envelope. Now the first layer, which is the glycocalyx, it is made up of mucus and polysaccharides, meaning higher complex sugars. Now, glycocalyx layer, it could be thicker in some bacteria and it could be thinner in some bacteria. That depends upon the type of bacteria we are talking about. So glycocalyx layer could be thick and it could be thin. And if it is like differing in thickness, it will also differ in the composition, meaning the thing like how much amount of mucus it has. Like maybe mucus is more, polysaccharide is less, polysaccharides are more, mucus is like that. So chemical composition for different bacteria, it would vary. Now the glycocalyx, it is basically of 2 types or of 2 ways it could be present. First is it could be present like a slime layer, and the second it could be present like a capsule. The slime layer is a loose sheath-like layer. It's like this is the glycocalyx. So on top of it, a loose sheath. Like if the glycocalyx is in the form of a loose sheath, then that is what we call as a slime layer. Okay, now see, slimy layer, it's like sticky. Okay, like how slimy stuffs are there. Now, why is it there? It's there in the bacteria to protect it from the loss of water. Okay, the sticky substance, the sticky layer, does not let the bacteria lose water. Whereas, if the glycocalyx is tough, we are going to call it capsule. Okay, and this is a thicker covering and it is tough in nature as compared to the slime layer. Now, this capsule, it provides sticky character to the cell of the bacteria, because you know that there are many bacteria that they have to attach to some sort of substratum. some sort of surface they have to
Hmm.
attach. Even though they are microscopic, still they have to attach to some sort of surface. So that is done with the help of capsule. So glycocalyx is clear.
Yes, ma'am. Clear.
After that, there comes the cell wall. Now, cell wall, it is a tough and rigid layer, and very famous thing, it is made up of peptidoglycan. What it does is that the first and the foremost thing that is performed by the cell wall in all those organisms that have a cell wall is that they protect the cell from bursting or collapsing. So if, like, let's just say they absorb a lot of water, the cell will not burst. Or if they lose a lot of water, the cell will not get collapsed. So to protect the cell from collapsing or from bursting, the cell wall is present. And in the case of bacteria, it is made up of peptidoglycan. Then comes the plasma membrane, which is the innermost layer, and this is the layer that from where the cell starts, right? And in no matter which organism you're talking about, whether you're talking about eukaryotes or prokaryotes, the plasma membrane is selectively permeable in nature. Meaning it can allow some stuff to enter and some stuff to go out, but not everything. And it is very similar to the plasma membrane of the eukaryote. So it's like not a lot of differences are there, but let's just say eukaryotic plasma membrane will be superior. It will be better as compared to the prokaryotic one. Now, one such thing which is present in the prokaryotic plasma membrane is mesosome. So what happens is that, for example, if this is a cell, so cell has plasma membrane surrounding it. So this lining, this limiting structure is itself a plasma membrane. Now, many a times this plasma membrane, it sort of invaginates like this. At one place, like this is normally how it's present. Sometimes it would form a depression like that. And that structure that is formed by the invagination of the plasma membrane is what we call as mesosomes. Okay, these are called as mesosomes and they are very important for prokaryotes. So the extension or the invagination of the plasma membrane forms special membranous structure which are known as mesosomes. Now, this could be present in the form of vesicles, in the form of tubules like this, and in the form of lamellae, like network-like structure. But the question comes along, why is it there and why is it important? So first, these would be important for cell wall formation. So whenever the bacterial cell divide, new cell has to be formed, new cell wall has to be formed. So the cell wall is formed because of the mesosome. Like it helps in the formation of cell wall. Second, DNA replication and distribution to the daughter cells. So when during the cell division process, when the DNA replicate, it becomes like a copy of it becomes, which goes to the either of the poles, and then the cell forms. So first, like for example, this is a bacterial cell. Initially, the DNA is here. Okay, when it has started to divide, the DNA will be starting to go like, it will start to reach the opposite pole. Okay, and after that, it will completely reach the opposite pole like this. And then the cell wall will be formed and two daughter cells are made. So this cell wall formation, this DNA distribution, these all are done with the help of mesosome. Clear?
Yes, ma'am.
Many a times the mesosome also help the cell to respire, to secrete stuff, and most importantly, to increase the surface area of the plasma membrane. Now, you've studied how convoluted structures, like how in our small intestine is like this, and it helps to increase the surface area. So, same way, the mesosome is also helping to increase the surface area of plasma. Clear?
Yes, ma'am. Clear.
After that, there comes Gram staining. Now, see, Gram stain is like a color. Okay, it's like a dye. Okay, so what happens is that whenever we want to identify the different types of bacteria, okay, if I say what are the two groups of bacteria, what are the two types of bacteria? So you'd say, ma'am, gram positive bacteria, gram negative bacteria. So what is gram? See, it is named after Christian Graham, who developed this technique. And it is a special technique with the help of which you can identify or classify the bacteria into two groups. Okay, gram positive, gram negative. Now the bacteria that will take up the gram stain, like if you stain that bacteria and the bacteria will get colored or it will take up the stain, that bacteria will be called as the gram positive bacteria. But there are bacteria that if they are not able to take up the stain, so we call it as the gram negative bacteria. But the concept behind this is that, for example, you have seen how the cell wall has peptidoglycan. So there are some bacteria whose cell wall is very thick because they have a lot of peptidoglycan present in it. There is a higher concentration of peptidoglycan. Whereas in some bacteria, there is like a thin cell wall because of lower concentration of peptidoglycan. Okay. So those bacteria that have a thicker cell wall and they have more peptidoglycan, they take up more stain because they have more space for the colors to bind and to get absorbed. So they become gram positive because they take up more stains. And the ones that do not take up the stain, we call it as the gram negative because they don't have enough space to retain the stain. Clear? So that technique
Yes,
is
ma'am.
called as gram staining. And always remember that the mesosomes are only found in the gram positive bacteria that have a thicker cell wall. In them only mesosomes are found. Clear?
Yes, ma'am.
Any doubts?
No, ma'am.
Okay. Now, depending upon whether the bacteria has a motile structure present or it doesn't have the structure present, the bacteria, they are of two types, motile, non-motile. If they are motile, they have a flagella present. What is a flagella? It is nothing but if this is a bacterial cell, so a tail-like structure will be present, which will help the bacteria to move, to locomote. So that is called as a flagellated bacteria. So flagellated bacteria are the motile bacteria. Now there will be bacteria that do not have any flagella. So they will be non-motile, meaning they can't move. But the motile bacteria can move along. And these flagella, they are the extensions of the cell wall. So like normally, if this is the cell, surround like plasma membrane is there and then the cell wall is present. So from the cell wall, the flagella would arise because it is an extension of the cell wall. Clear?
Yes, ma'am.
Now the flagella, they are long and filamentous structure, which is made up of three parts: filament, hook, and basal body. So if you see the structure, this is how the flagella sort of looks like. This long part that actually comes out of the cell is the filament portion. The part which stays attached to the cell wall area is the hook, and the basal body is present even beneath that, which sort of from where the actual flagella begins. So filament is the longest part. It projects outside the cell. Is it clear?
Yes, ma'am.
Now, flagella is the extension of a cell. Extension of a cell means something that comes out of the cell. Same way, there are other structures also like pili and fimbriae. But these two structures, they have no role in helping the bacteria to move. They have no role in motility. They are there for different functions, but their origin is from the cell itself, like the cell extension. Okay. So when we talk about the pili, so they are tube-like structures, so tubular structure that are made up of the protein pillin. Okay, pillin makes the pili. Then there is fimbriae, which is small bristle, like how we have bristles of our toothbrush. So same way, fimbriae are like that. And what it does is that it helps the bacteria to sort of attach onto the substratum or the rock or wherever it is present. So for attaching purpose, fimbriae is there, and mostly in the reproduction, pili performs. Okay, like when the bacteria has to transfer DNA, which is sort of like a reproduction in that. Is it clear? And see,
Yes,
fimbriae
ma'am.
looks like this. Bacteria cell having several bristle-like structures, which we call as fibrin. Clear?
Similar to cilia.
Hmm?
Similar to cilia.
Yeah, similar to cilia. It's just that cilia has no role in attaching anywhere. Cilia has role in propulsion, like it will propel things like this. Like how windshields are there, like that. But fimbriae, they have no role in propulsion of anything. They don't move anything forward. They help the bacteria to attach onto something. Clear?
Yes, ma'am.
Any doubts?
No, ma'am.
Then after that, there comes the prokaryotic ribosome. Now, prokaryotic ribosomes in general, whether prokaryote, whether eukaryote, it is responsible for the formation of proteins. They are the protein factories, and proteins are very thousand times very important in any of the cell, because most of the things that are formed in our cell or in our body, they are proteinaceous in nature. Okay. Now, the difference between the prokaryotic ribosome and the eukaryotic ribosome is the sedimentation coefficient. Okay. So first you note down till here, then we'll move forward. Done, Shimla.
No, ma'am.
Okay.
Done, ma'am. Done, ma'am. Done, ma'am. Done, ma'am. Denmark. 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 Done, ma'am. Done, ma'am. Done, ma'am. Done, ma'am. Done, ma'am.
All right, any doubts up until now?
No, ma'am. All clear.
Now, see, what happens is that this ribosome, it is actually of two different types, 70S and 80S. 80S is the ribosome of eukaryotes, whereas 70S is the ribosome of the prokaryotic organism. Now, the size of a prokaryotic ribosome is 15 nanometer by 20 nanometer. That is its size. Now, these ribosomes, they are particularly associated with the plasma membrane. See, sometimes the ribosomes are present in the cytoplasm. They are present on the endoplasmic reticulum, and they can also be attached onto the plasma membrane. So here, in the case of prokaryotes, the ribosomes, they are attached or associated with the plasma membrane. Now, the type of ribosome found in the prokaryotes is the 70S type here, and here S stands for the sedimentation coefficient. Now, the 70S, like every ribosome is made up of two subunits. One would be a larger subunit and one would be a smaller subunit. So the larger subunit of a ribosome is 50S and the smaller subunit of a ribosome is 30S. Okay. Now, in the case of prokaryotes, what happens is that several ribosomes, they come and they attach themselves on the mRNA. Okay. And this structure, this association sort of looks like this. This is this. This is the mRNA. Onto them, the ribosomes will come and attach. So this structure that is formed by the attachment of the ribosomes onto the mRNA is called as polysome or polyribosome. What are they there for? So they are helpful in the process wherein from the mRNA, the protein has to be made. So for that, for the translation process, they are very important. So I'll write it over here for translation. And what is translation? Translation is a fancy term for protein synthesis. Clear?
Yes.
Understood?
Ma'am, what's the M in RNA?
mRNA, see,
The
in
M.
our body, DNA and RNA both are present, right? DNA and RNA, they are called as the nucleic acid. Now, RNA, RNA stands for ribonucleic acid. Okay, and RNA is of multiple types. mRNA, rRNA, tRNA. Okay. So this M stands for messenger. So this is the messenger RNA. R stands for ribosomal RNA. This is the ribosomal RNA. And the T stands for transfer RNA. So these are the different types of RNA. So the ribosomes, they particularly attach onto the mRNA for the translation process. Clear?
Yes, ma'am.
Any doubts?
No, ma'am.
After that, another structure that the prokaryotes have is the inclusion body. Now, these are spherical structures that are non-membrane bound, meaning they do not have any membrane surrounding them and they are just lying there in the cytoplasm of the prokaryote. Now, they imagine them like the storage structures of the prokaryotic organism. So, the reserve food material of the bacteria, they get stored in the form of inclusion body. So, it's like the main purpose of it is to store stuff. Reserve food material, sometimes different granules are there. So different granules like phosphate granules will be storing phosphate, cyanophycean granules, glycogen granules. So these are all the different examples of the inclusion bodies that are there in the case of prokaryotes. Is it clear?
Yes, ma'am.
Okay. Then comes the blue-green algae. The blue-green algae is like sort of like a group that comes under the prokaryotic group, which is like just like bacteria only, but they have this capability of this thing, photosynthesis. Okay, like they have certain pigments in them with the help of which they can perform photosynthesis. And there are also purple and green sulfur bacteria. Right, you like how you have studied in biological classification, the cyanobacteria and mycoplasma, all of those things are there. So they contain the gas vacuoles. Why? Because they are aquatic and the gas vacuole helps them to float. Clear?
Yes,
So
ma'am.
this was all there is to know as far as the prokaryotic organisms are concerned. Because they are not very superior, so they don't have a lot of surface structures and stuff. This is what they have. Okay. Now we'll move on to the eukaryotic cell. These are the ones that actually have highly complex structures present in them. They have, you can say, better adaptive structures. They are superior in every way and every form. So the kingdom Protista, fungi, plantae, animalia, all of them are eukaryotic organisms. They could be unicellular, but all of them are eukaryotes. Now the cells that have a proper membrane bound nucleus, that have a proper like membrane bound cell organelles, we call them as the eukaryotic cell, right? And in them a proper plasma membrane is also there. Now cell wall is present, but it is present in the plants, fungi and the protists. It's not present in the animal cell. Majorly, let us see the differences between the plant and the animal cell. Now see, the plant cell as compared to the animal cells, the plant cells are going to be larger. The animal cells are going to be smaller in size. The plasma membrane of the plant cell, it has another layer, an extra layer is present, which is rigid in nature, and that is the cell wall, which is made up of cellulose. Whereas this thing is absent in the animals, they just have plasma membrane as the outermost layer. That's it. No other layer, none. Not even a single layer other than the plasma membrane is present. Now they are plant cells, they have plastids, right? The chloroplast, chromoplast, leucoplast, different plastids are present or variety of the functions. But here, no plastids, none of the plastids are present. Most mature plants, they have a permanent and a large central sap vacuole. Now see, plants, they have vacuoles in them, whereas animals, if they do, they are temporary vacuoles. Many a times the vacuoles are absent. So, clear? Vacuolar
Yes, ma'am.
structures, they are mostly found in the case of the plants. Then the Golgi apparatus of the plant cell, like Golgi apparatus is another cell organelle. It is simple and we call it as dictyosomes. And in the case of animals, the Golgi apparatus, they are highly complex structures. Clear?
Yes, ma'am.
So now the plant cell, they lack centrosome and centriole. See, these are the structures that are involved during the cell division. Helpful in cell division. So they don't have it, whereas the animal cells, they do have. Clear? So these are the
Yes,
basic
ma'am.
differences. There is nothing else that you could write. Even if it comes for 5 marks, this is like more than enough. Okay. Like we have covered every single point. Now coming on to the plasma membrane, the cell membrane of the eukaryotes. So we know that it is the limiting membrane or a limiting structure that surrounds the cell, and it was discovered around 1950s. Why? Because at that point of time, the microscope was invented. So the people, like, they could understand the structure of the plasma membrane better by visualizing it under the microscope. Now, when like different people were studying the structures of microscope, sorry, not microscope, the plasma membrane, and people gave their own theories as to how they think the structure is. But then came two people, Singer and Nicholson. Then in the year 1972, they gave a proper structure of the plasma membrane. Okay, they gave a model, and they called the model of the plasma membrane as fluid mosaic model. Okay, so according to them, what they said is that the plasma membrane, it has a lipid bilayer, meaning it has two layers of lipid, and the proteins, they float in the lipid bilayer. Okay, this is what they said. And they also said that depending upon the ease of the extraction, like whether it is easy to extract the protein or not easy to extract the proteins, they are of two types: integral protein and peripheral. Integral means it's like very deeply situated, so it will be difficult to extract it. Peripheral means it's present on the surface, so you can easily extract it. Clear? This is like the basic thing that they said. So according to the fluid mosaic model, which was given by Singer and Nicholson, the plasma membrane was said to have a quasi-fluid nature or semi-fluid nature. Okay, why? It's because of the lipid that helps or enables the movement of protein. So if this is like two layers lipids are there like this, and the protein moves to and fro in the lipid bilayer. Clear? So
Yes, ma'am.
yeah, so lipid is present in a bilayer structure. Now the function, the fluid nature of the plasma membrane, it is very important because the cell has to grow, it has to expand. So the plasma membrane will also expand accordingly. It is helpful in cell division process, secretion process, and also in the endocytosis. What is endocytosis? It is like the whole entire process wherein either the food or the liquid particle are taken into the cell through the plasma membrane. Clear?
Hmm.
Do you remember pseudopodial movement, how that happens? Like the amoeba draws out the pseudopodia, takes the food and eats.
Yes, ma'am.
So that was because of the plasma membrane's fluid nature, semi-fluid nature. Okay. And then
Yes,
same
ma'am.
thing comes, like the opposite of the endocytosis is the exocytosis, which is also called as cell vomiting or ephagy. So it's like whenever the food or any particle that is now used up, that gets thrown out or excreted out of the cell or out of the cytoplasm of the cell through the plasma membrane. So it's like expulsion of the particle. Endocytosis is intake of the particle. Clear?
Yes, ma'am.
Now the plasma membrane, it allows the transport of materials across it. Why? Because it is selectively permeable in nature, it can allow stuff to pass through it. But it's not that it will allow every single thing to pass through it. It will be selective in nature. It will see which things can be passed through and which cannot. Okay, so after that, there comes the different types of transportation that happens across the plasma membrane. Now, what is the basis of studying these types? The basis is that whether the transportation process requires energy expenditure or not. Okay, passive and active transport are the two types of transport. Now, passive transport, you know that, for example, if I give you two situations, okay, tell me in which one would the molecule pass through easily? If we go from higher concentration to lower, or if we go from lower concentration to higher, which one would be easier?
Higher to lower.
Higher to lower, meaning across the concentration gradient, along the concentration gradient, isn't it? Whereas low to high means against the concentration gradient, isn't it? So passive transport is the transport that happens along the concentration gradient. It does not require any energy expenditure. So it takes place along the concentration gradient, meaning from high to low. So it is the movement of molecules without any energy requirement. It just requires a gradient, meaning somewhere something has to be at higher and somewhere something has to be at lower concentration, and then only the molecules will move in this case. So what the only thing that is needed is concentration gradient and nothing else. If the concentration gradient is there, naturally the thing will pass. Okay?
Yes, ma'am.
Then comes diffusion. Now you know already the movement of gases from high to low concentration through the semi-permeable membrane. So this is an example of passive transport. Doesn't require any energy. It happens along the gradient. Okay, and when the same thing happens in the case of water, instead of calling it diffusion, we call it as osmosis. So the movement of water is called as osmosis. Neutral solutes. Now, which molecules can do this passive transportation? It's like those molecules that do not have any charge on them. Like they don't have a positive charge, neither do they have a negative charge. So the neutral molecules, the neutral solutes, they will be doing passive transport. They will not require any energy to pass through the plasma membrane. Is it clear?
Yes, ma'am.
Any doubts?
No, ma'am.
After that, there comes the active transport. Active transport or uphill transport. It's just like how if you're standing at the bottom of the mountain and you want to go up the hill or up the mountain, that would be difficult as compared to coming from the mountain to the bottom, right? So active transport is the transport that takes place against the concentration gradient, meaning it takes place from low concentration to high concentration. So naturally, it would require energy because if you think about it, when you move from the top of a higher place to a lower place, it's easier to come down, isn't it? We don't even have to put any effort. We just come along it. But when you're climbing up the stairs or when you're coming down from going up the mountain, then you would require energy, isn't it? You require strength. So same way, in the active transport, since it is happening from the lower concentration to the higher concentration, so it requires energy, and which is obtained from the energy currency of the cell, that is ATP, which stands for adenosine triphosphate. Okay. If I say adenosine triphosphate, then how many phosphates does it have? See the name? See the name? Adenosine triphosphate. So how many phosphates?
3.
3 phosphates, yes. ATP is made up of 3 phosphates. Okay. So this active transport, it is an energy dependent process. Now, which type of things or molecules would pass through? The ones that are charged, like sodium, potassium pump is an example of active transportation. Is it clear?
Yes, ma'am.
Any doubts up until now?
No, all clear.
Oh, very good. After that, there comes the cell wall. So the cell wall is found in plants, it's found in bacteria, and it is found in the fungi. Animals do not possess a cell wall. The plant cell wall is made up of cellulose. So we call it as the cellulosic cell wall, right? And this cellulose is a complex sugar. It's not protein, it's not anything else, it's a complex sugar. Now, apart from the cellulose, the cell wall of the plants, they also contain hemicellulose, pectin, and different kinds of proteins. But the major component of a plant cell wall would be cellulose. Then comes the fungus. You know already that the fungal cell wall is made up of chitin. So we call it as chitinous cell wall. Then comes the algal cell wall, which is made up of cellulose, galactins, and mannans. Okay? Any doubts?
No, ma'am.
Okay. Now see, the plant cell wall, it has two regions, primary wall and secondary wall. See, when the cell wall of the plant is being formed, the first layer of the cell wall that is formed is called as the primary cell wall, the first formed cell wall. And later on, the second time the cell wall layer will be formed, that will be the secondary wall. Okay, and in between the primary and the secondary, cementing material will be present, which will be called as the middle lamella. So the primary wall, it is the first formed cell wall of plants. It is thin and expandable. Most of the young plant cells, they just have a proper primary wall. They do not have a secondary wall, the younger plants. But when the plants mature, in that case, new layer is formed after the primary wall, which becomes the secondary wall, and it is thicker. Okay, it is thicker than the primary. Then the cementing material that is helping the primary wall and the secondary wall to stay together, that is the middle lamella. And what is it made up of? It is made up of calcium and magnesium pectate. This is very important. Like, what is it made of? Many a times they ask. Then comes plasmodesmata. See, in the plant, like, and normally in any kind of tissue or any kind of organism, there are several cells present. So how does one cell connect with another cell? So in the case of plants, they connect it with the plasmodesmata. So plasmodesmata, they are the strands, like cytoplasmic strand, that connect the two adjacent cells together for communication. So it's like a tube-like structure or a strand-like structure in between two cells, helping them to communicate. Clear?
Yes, ma'am.
Now talking about the variety of the functions of the cell wall. So first of all, the cell wall is a rigid layer. So it provides a proper shape to the cell. Proper shape. Second, it protects the cell from mechanical injury. It doesn't let any kind of injury take place in the cell. Then it helps the cell to, you can sort of protect itself from the pathogens like bacteria, viruses, and fungi, because it acts like a barrier. It doesn't let these things come inside. It allows the material to pass in and out of the cell, and it helps in cell-to-cell interaction. And obviously, if any cell has cell wall present, so that cell is not going to burst, nor will it shrink or collapse. Is it clear?
Yes, ma'am.
All right. Any doubts? Surely clear it is up until now.
Yes, ma'am. Yes, ma'am.
This chapter is not difficult. It's just that it's lengthy. So you have to learn a lot, but it's not difficult. Okay. So you were
Yes,
writing,
ma'am.
you were writing ribosomes, right?
Yes, ma'am.
Okay. Now you can note that.
Done, ma'am. 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 Done, ma'am.